So what we did for the group, including Dick, about 2, 3 months ago, is I talked more about a proposal we were putting together for the EU, an EIT proposal, to bring this kind of approach out to beyond where we're using it currently in the Netherlands, where I'm based. I'm Matthew. I'm what's called a structural biologist. Some of you may have seen in the news recently about AlphaFold 2. It was a big noise, at least for us in our community. So Google have essentially destroyed my science by being able to predict protein structures for all human proteins, which is fantastic. It's really going to change how things work. But it's just the start. We can still use those structures to try and help clinicians make the right choices for patients who are undergoing therapy. And that's something Google doesn't do yet, but we do and have been for the last 5 years. Unfortunately, Dick, the EIT funding, we missed by 0.2% of a point. We scored the highest in the Netherlands, Luxembourg, Belgium, and Israel combined, but we still didn't get funded. But I don't think that means we have to stop here. We're going to continue at least. The title of this is essentially how we, as people who understand what proteins look like, can help the clinicians who are treating you, treating other cancer patients with their specific diseases, making the choices between what kind of medications. So in the field of lung cancer, there's a large number of medications, and as you're fully aware, trying a patient out on a medication to see how he or she does isn't really the best way forward. It's much better if we can make a knowledge-based decision. Can I move forward here? Yeah. So we've been doing that since around about 2016. So shockingly, about 5 years ago. My head of department's boss came to me and said, there's a group of clinicians who meet at the UMCG, which is the University Medical Center in Groningen. That's Groningen, but pronounced in Dutch. And they would love to have somebody come on board and join their meetings about patient treatments who understands what proteins look like on a molecular level and what the drugs that are being used to treat the proteins look like on a molecular level. And initially, I have to confess, I was a bit cautious about climbing out of my ivory tower and getting involved with the real world. And it's been massively rewarding, challenging, massively rewarding over the 5 years. Because what I learned was that these clinicians who are treating patients, they are, undoubtedly experts, and they are deeply gifted and motivated people. But when it comes to understanding what the medications they're giving to patients are doing on a molecular level, they only get about 2 hours of training over their medical degree. So they simply don't really see it. Certainly not in the way that I see them. So what we found was that the way we're treating, or the way we're helping the clinicians at the EMCG treat their patients' diseases, it kind of springboarded out, and we started getting calls from Amsterdam, from Leiden, from Leeuwarden, from Antwerp, from Quebec, from Paris, as more and more people heard about what we were trying to do and said, well, can you help us with our cases too? Which was a bit of fun, because at the beginning it was me on my coffee breaks trying to pat away on a keyboard and come up with solutions. And then we were lucky enough to get a PhD student funded for 4 years. And now we've recently had another one join the group. And since then, we've been able to spring out and handle more and more. And there's a lot of, from my cynical point of view as a scientist, there's a lot of really interesting questions to answer here, as well as trying to impact patient lives. The direct impact is felt predominantly by the insurance companies, because over here, they're the ones who bear the cost of the treatments. So if we are talking about choosing between two different tyrosine kinase inhibitors for targeted therapy, then there are big savings to be made by choosing the right course of medication rather than trying the patient on the wrong course for 2 weeks and then changing them onto a different course for 2 weeks and then onto another course for 2 weeks before they can get treated. So there's a big saving to make financially for the healthcare insurers. Of course, there's a time saving for the patients to put them on the correct therapy as early as possible. And there's also an impact on ensuring that we're not giving medications to patients unnecessarily. So when we do what we do, I can predict even potentially when the medication simply won't work, and perhaps a targeted therapy is not good for this patient. They should be looking at other alternatives, chemo or radio or immunotherapies. Yeah, and that will therefore impact their side effects and their quality of life as they're going through their treatments. And we now have, over the last 5 years, I think 480 cases that have gone through this group of experts that we joined, the molecular modeling, sorry, the tumor board. And the best indicators I have at the moment is that I talked to Harry Groen, sorry, 2 months ago, and he said the result after the 5 months of the tumor board is that at 6 months, the survival of these patients has improved from 50% to 80%. So that's an N is 480 case. And I think that's a real world difference we've been able to make. These are, admittedly, these are quite ill people to start with, but we're quite proud of that as an outcome. Our goal with this is within a 2-week window to be able to use a computational approach to be able to choose between expensive treatments. But that's not really the most important part, because we would rather also help patients understand why they're being asked to take this medication. I mean, as I'm sure most of you are aware, you don't feel great taking these medications, and you want to understand why you're taking these medications, to understand, to maintain the dosage, to keep on track. And I think some of why you're taking it is not clearly understood, whereas a picture will show 1,000 words. And as I stop wittering on, I'll start showing you some pictures and you'll see more and more about how medications are actually working here. It'll also help reduce the stress and the workload on the healthcare professionals, which I think is then going to give them more time to talk around and find different things. This is essentially what it boils down to. So as I said, I'm a structural biologist, which means to me, this is a readily interpretable image. Okay, this is something I've been doing for around about 20 years. Depending on how deep you've gone into your own therapy, this is something you may be something more familiar with. So when an oncologist does a gene panel for a patient, they'll get a DNA sequence out for you. Okay, and that DNA sequence can help drive their decisions as to what is driving your disease, what kind of therapies are possible for you. Here, in the case of lung cancer, many cases are driven by EGFR. It's a protein, one of 20,000 in your body. And in this case, this hypothetical case, the normal patient, the healthy patient, will have a sequence of DNA that then is translated into this amino acid sequence. And this is the dogma of biology. DNA becomes RNA, becomes protein, and it's the proteins that do what the job is. So in this case, the wild type sequence is referred to it. The healthy EGFR sequence will be this selection of letters, okay? And a patient may present at the hospital with this selection of letters. And there's only a minor difference here, but this is enough for that patient to start showing disease. But this is not the truth. This is not really what's going on. No more than A, T, C, and G are the truth. A closer approximation of the truth is that these letters form a chain of atoms, amino acids, that fold up in sequential order to give you an overall shape like this. And it's this molecule here, it's this shape of this molecule here that gives its function in the human cells and then causes progression or uncontrolled growth of the cells by interacting with other things around it on a molecular level. And what we can do, because we understand structural biology, is we can say, well, this minor change, this C to an S, this is the simple addition, the simple swap of a sulfur atom for an oxygen atom. That's all it is, which means as we know what the wild type looks like, we can create a very good computational model of what's driving this patient's disease. Now, in the early days of this project, we were predominantly using this to help decide between which way a treatment course for a patient should go. Somebody was showing resistance to afatinib, a TKI, a therapy they were under, and the clinicians were saying, well, is there another one we can use? The literature doesn't suggest anything to us because it's a rare mutation. We've never seen it before. What do we do? And actually, because we can model it on this molecular level and we know where all the atoms are, as I said, I'll show you this in a more three-dimensional representation so you can see things more clearly. We are able to say, well, okay, afatinib, it won't work for this patient. Why? Because this sulfur has become an oxygen and that the chemistry no longer works for that. And the medics are going, okay, explain this. So you show them here, the bonds aren't made in the right way. Afatinib won't interact with the protein in the right way. You want to give this patient osimertinib, and that should work. Okay, and by and large, these are the right kinds of outcomes. And we see this as we've gone through our work. If we go deeper into the science, which I said, from my cynical point of view, this is something I'm also interested in, we can start to also predict how strong individual binding interactions may be, which starts to be able to give us an idea about dosages. Yeah, so not everyone needs 250 milligrams of compound A. Some people need more, some people need less, because some people are different, and we can account for those. We can also start to talk about selectivity, because when we see this shape here, this is the EGFR protein that's driving this patient disease. Well, this is one of about 1,000 different kinases that are present in a healthy human. So we can also create models, thanks to AlphaFold 2, of the other 999 kinases, and start to ask questions of, well, if we give this patient, him or her, afatinib, how is it going to interact not just with the target protein, but the rest of the kinases in their body? Is it going to have too many what we call off-target effects? Is it going to interact with too many other kinases that are essential for their normal healthy progress and give them worse side effects than is necessary? So we can start to talk about the selectivity between. And ultimately, our aim is to give a knowledge-based estimate of which drugs are going to be used and not rely on A's, T's, C's, and G's, or some kind of alphabet soup that doesn't really mean anything other than as text. This was our first story. So this was a patient who was showing progression under A treatments in Quebec. And essentially, she had progressed along this treatment course, and it was currently at that point on the latest therapy that's available for non-small cell lung cancer, osimertinib. And she was showing progression on that. And unfortunately, the way the books are written is that you start off with the first line therapies and you move on to later line therapies. And at this point, the clinician said, well, we're out of therapies for this patient. What's the next step? But because this patient was sequenced and we could see that they picked up another change in their alphabet soup, actually what we could say is, well, don't worry about a fourth line therapy that doesn't exist. Go back to this first line therapy. This one should now work again. And indeed, in this patient's case, that indeed resulted in a decrease in her tumor mass, and she did very well for a while. Limitations here. Your faces are on the right of my screen here, so can you actually see the whole of my screen, or are you blocked?
No, it's good.
You can see it. Great. So what we are limited by is we're only 3 people running this project at the moment. Yeah, but we want to expand it. And that's one of the reasons I'm talking to you guys here, because everything I've talked to you so far is about non-small cell lung cancer. But of course, the kinases that drive non-small cell lung cancer are very similar to those that drive other cancers. Cancers and kinases are intimately linked, and they behave in exactly the same way, and they behave in the same way in our computers. So of course, we can look at other kinases and other models. We've increased our database, but we haven't made it globally active yet. And there is a kind of a choice we have to make in the time window. So, of course, computer time, it depends on the computer you have, but to model these kinds of things is a little bit exhaustive on resources. So we do have to make a choice between how complex we model the situation and whether we try and take a quick result to be able to give a solution for that patient. Here, we're not trying to come up with a solution for patients in the future, but we have a 2-week window essentially where we're trying to give an answer to the clinicians. We have our regional collaborations, and we're trying to identify people to be involved with this. And that's one of the reasons I think that Dick put us in touch with you guys. I'm a scientist, so here are my 4 recent publications on the area where you can see what this kind of approach has made, or the difference this kind of approach has made for non-small cell lung cancer patients. And we're currently working on a few more ideas, which I'll get to. Our current thinking, as I said, is to move away from this ATGC or this alphabet soup of sequences and actually allow clinicians to be able to use these models to choose between the right medication for the patients. But of course, the same is also true on the other side. Yeah. As long if the patients start to understand what these mutations mean and how it can inform their treatment choices, they will be better empowered to understand their therapy, take part in their therapy, and also bring in more clinicians for us to start to communicate with. So the whole thing hopefully grows out. those are my contact. I'll come back to that later. So that was a lot of words. And I think the easiest way, hopefully you can still see my screen. Yes? Is to actually give you a couple of live examples. So this little beastie is something we were looking at earlier late last week. This is a three-dimensional model of the kinase driving this patient's disease or patient's disease in lung cancer. This particular representation is an experimental model. So it's been solved. That's what it looks like to within experimental errors. And the experimental errors, trust me, are quite small. You get a relatively intricate image, but what you're seeing there is the line of all the little amino acids joined up. Here's the amino acid code here. So if I select a bit of this, you can see that's how the amino acids are joining up. And they form this very long, intricate structure. Okay? And the whole purpose of this intricate structure is nothing more than to bind, well, two molecules. One molecule of ATP, which is the energy workhorse of the cell, because it allows phosphates to be transferred onto things and then creates other molecules talking to other molecules. And also, in this case, to allow the EGFR to put that phosphorylatable molecule, this phosphor residue onto a specific amino acid of its target protein. EGFR interacts with a variety of different molecules in the human cell. And unfortunately, if it's misregulated, it starts switching on too many pathways, promoting cell growth, and then you get, tumor growth. What we have in this case are two things. So the first is from an earlier patient, there was a specific mutation this patient picked up, which was a glycine at this position. That's the normal form. And if I can zoom in on that, hopefully I can zoom in, and I should center on it as well. Is it centered? Yes, it claims it's centered. So typically that's a glycine amino acid. If you come and we set up the MOOC course, you'll understand more what a glycine is. But the key is that in that patient, that glycine had become an alanine. Can I get that to show pop up? Okay, and now I have to zoom in. And what's happened there is the glycine residue doesn't contain one atom there. That's the only difference between the glycine and this new residue, an alanine. And this new atom from the alanine pokes out here into this pocket. And not only is this representation I'm showing you there, the EGFR, it's also showing you lapatinib, which is a known therapeutic for EGFRs. It's a known inhibitor for patients. And what I can tell you is the moment you add this atom here, it gets too close to this. And two atoms cannot occupy the same space, basic chemistry. So that means this atom means the lapatinib can no longer bind. So what we need to do is look for a molecule that can still bind. And for that, we can find, for instance, osimertinib. Here is a similar structure. Again, this is EGFR. Don't mind the color change. Here is our osimertinib molecule bound down there. And if I can find it correctly, 719 is that one. Here is my alanine. And you'll have to take it from me, but the distance between these two is just large enough to not inhibit binding of osimertinib. So in this case, we're able to suggest for this patient who's showing progression on afatinib, put her on osimertinib, and she should respond to this because I can see it from where that atom sits. The other thing that was quite thrilling, again, for me as a scientist last week, was we were asked a different question. So we weren't asked questions for patients who are already being treated for an EGFR-driven disease, because in those cases, we already know they're under treatment with afatinib or gefitinib, and they showed response, and then they started to show progression of disease. So we know what the molecule behind their disease is. It's EGFR. There's a different question here, however. So on first presentation, so early stage sufferers, is it EGFR that's actually causing the disease? And for that, the example that was given to us was this 703L. We talked about that last week. And we'll leave that alone because it's still, no, that should be phenyl. So Leon, who we work with, he's a molecular clinician, a molecular pathologist, said that he had a patient turning up with no previous treatment, of course, at all. And they found a new mutation on her EGFR again, which was a leucine residue to phenyl residue at this position. And what was this? Was this potentially something that meant the EGFR was driving this disease? And that was something that up until last week, I wouldn't have been confident to answer. But now Lily, who's working with me, and I've actually had a look through a number of the basic features of this molecule, and this confirmed one of my hypotheses. So there is a lot of mechanics behind this, but what you're seeing in this model is what's called the inactivated form of EGFR. And it's inactivated because this little beastie here, which we call the C helix, actually sits close to the surface of the protein. It's kind of docked in like this, and it closes off access to where all the machinery of this thing works. And also helps this thing here, which is the inactivation helix, bind. So what we found by looking at a library of activating mutants is that almost all these mutants cluster in this region and play in our models with how firmly this element of the EGFR binds and how firmly that then binds this inactivation helix. And that allows us actually to say, no, these are not just random mutations you can ignore because the patient is different. These are not these are not mutations that are labeled as activating in literature, but actually you'll find these are activating mutations. We're fairly confident. Here, in this case, you can see in the red circles there, actually it's showing some of the chemistry for you, and it's saying when this is now a phenylalanine residue, so it's larger and bulkier than a leucine, it actually starts to make too many clashes with the rest of the protein, and it's labeling those up with red. So that was a 20-minute tour of protein structures mixed in with drug design, mixed in with some quantum mechanics, mixed in with some old school chemistry, and it is woefully incomplete. Yeah, but I hope you managed to get an impression of what the proteins are doing and how we can use this information to hopefully improve patient treatments. I'm going to close Pymol now, and classically when I teach online, that means my screen goes dark. So please tell me if my screen's gone black. I'm still here?
You're still there.
Excellent. And
Yep.
No, wrong presentation. That's you. So you can still see me?
Yes.
Yes.
Okay. So for the recording, if you want to follow up, that's my email. Drop me a line and we can chat a bit more. That's my website, and that's a very old picture of me because I now look a bit grimmer. I would dearly love to talk to any medical or oncologist contacts you have to bring them into our loop. So yeah, at that point, I would stop.